today we are going to be discussing our project which focuses on sun plants versus shade plants. So let's jump right in. We chose to compare how the leaves of plants growing in the shade differ from those that grow in the sun. We chose to do a total of 10 plants. We chose 5 plants growing at the Arboretum to view our sun variety and 5 plants growing in the woods behind Jack Baskin as our shade plants. Our hypothesis was that the stomatal density of sun plants will have higher water loss stress. Shade growing plants will contain more chlorophyll due to them getting less sunlight. And the parameter will reveal that sun-growing plants will have more photosynthetic activity than shade plants. To test our hypothesis, we chose the apogee chlorophyll meter to compare the levels of chlorophyll and the Decagon SC1 parameter to measure water loss, and as well as nail polish to make impressions of the epidermis to measure the stomatal density. We chose to study plants that grow in varying light intensities to compare and contrast different species of plants that grow in similar conditions.
The sun plants we used, we went to the Arboretum and picked out 5 plants. Going from left to right, the sun plants we used were the golden pagoda, the yellow conebrush, the wagon tree, the protea pink ice plant, and the proteus. And for the shade plants, we went in the woods behind Jack Baskin. And from left to right, those plants are the tennoic, shield ferns, trilliums, red alder, and then wood ferns.
We took clear nail polish with us to the Arboretum and to the forest behind Jack Baskin and used ImageJ software to measure each plant's stomatal density. However, I do need to put a disclaimer that our slides had a lot of air bubbles, and the data that we collected is pretty skewed and is not the most accurate representation of sun plants versus shade plants. Here are some examples of the images that we were able to achieve through ImageJ. The center and rightmost photo show that there are a lot of air bubbles on our slides. 2 big issues that this could be causing is covering up some of the stomata and just creating issues for focusing. We weren't able to focus on the entire slide. It was focusing on the right side or the left side and making it really hard to truly come up with these calculations. According to the images that we were able to produce, the shade plants had a higher stomatal density than the sun plants. However, it is important to note that we had a harder time with imaging these shade plants, and that could have skewed the data.
For the chlorophyll meter averages, in sun plants, the averages were 58.7, 19.6, 36.2, 43.3, and 40.5, versus in shade plants, it was 23.8, 25.5, 32.6, 11.5, and 16.3. For the parameter averages in sun plants, it was 187, 157.5, 160.1, 245.7, and 362.3, versus in the shade plants, it was 187.3, 133.3, 165.5, 90.9, and 76.5.
Our findings helped us compare our results to our hypotheses. Our hypothesis for stomatal density was incorrect. Shade plants had a bigger stomatal density than sun plants, which yielded an average of 2.21 times 10 to the negative 4th. Our hypothesis for the apogee chlorophyll meter was incorrect. Sun plants had more chlorophyll content, yielding an average of 39.62. Our hypothesis for the Decagon SC1 parameter was correct. The sun-growing plants had more photosynthetic activity than shade, which yielded an average of 222.54. Thank you for listening.